Okay, so I had to do this again. I recorded a good portion of it, so I'll explain what I wrote down here just so you can see. So, so this part here, first of all, DNA is the blueprint, the main point of this. And we need to go over what we mean by chromosome. We talked about that in grade 9. A chromosome is just a condensed form of DNA. And human beings have 23 pairs of chromosomes. Species more in a chopped up form and by proteins. Okay, so what are genes? Genes are sections of the DNA. So I was putting them out here, here. Those are little genes, and they carry the code for a particular protein. I'm going through this a little faster because I did do it again once. Hopefully, anyways, but if you're finding I'm going too fast, you can go over it again and just try to catch up to where I left off. A gene consists of a sequence of thousands of DNA base pairs. Go back to the last lesson that I gave on base pairs on the DNA. And the genes act in pairs that dictate what we call traits or features or characteristics, like hair color, like skin color, like the size of the nose, and or how good your eyes are, or I think I already said eye color, but eye color, your height, your body structure. Okay, so let's go to traits here. They are characteristics organism can pass on to its offspring. Who is the actual discoverer of what we now call modern genetics? Another religious, is a monk. So we talked about this before. It's a common fallacy that claims that there's a contradiction between science and faith. Actually, most of the earliest pioneers were faithful in some way. They were either of a religious order or they were laymen or women who, in most cases, had a very strong, not because they were a product of their times, but they tended to be very religious in nature. So you'll find that if you know the history of science, that's an actual fact. It's been glossed over. So, little known fact. Gregor Mendel was an Austrian monk, experimented with garden peas, and developed the foundation of what we now call genetics. What did he observe? He noticed that peas had several traits or features or characteristics, and that they all showed up. Only one or the other, rather than a blend, which is what previously was believed. He crossed plants with different traits to see what the offspring would look like. So it was on one trait or another. In other words, if we look down here, we're going to again look at some of the results that he got when he crossed pea plants. So you either got a round seed or a wrinkle seed, or a yellow seed or a green seed, not a blend of both. And there was always one, in the case of shapes, one shape that was dominant. In the case of colors, one color that was dominant. Okay, so Mendel found, let's go back to here. Mendel found that no matter what combinations he tried, one trait always dominated, masked the other. In other words, the plants always showed one trait, not the other. They were either had round peas or wrinkled peas. It didn't matter if the trait came from the male or female. The traits were controlled by something he called factors, which were later called genes, which is what we've talked about, we mentioned previously. Okay, so these are all examples of different traits. Seed color, flower color, pod shape, pod color. Mendel his crosses between tall plants yielded only tall pea plants. So tall combined with tall only gave tall. Similarly, short only gave short. Okay, this is when he was working with pure plants. The tall would only have the gene for for tall plants, and the short plants would only have the gene for short plants. Okay. And I kept doing this over and over again so I could make sure that these were pure tall plants with only those type of genes and pure short plants. And then he crossbreeded them to see what happened. So he breeded a tall with a short. And what he ended up with, he found that he ended up, where I left off here, he ended up with tall plants. And this is called F1, or the first generation. Okay, we go back here. These are all called the parent generations. These are all parent generations. Okay, parent. That's a parent. That's a parent. So when you blended two different parents, you got the dominant trait showing up. That's all. Okay, what about what happened when he blended two of these tall, two of the first generation? Okay, so this here and here is this one here, the first generation plants. So he blended two of the first generation plants, which were tall. Okay, what did he get? He ended up getting, this is the offspring here. My pen's not ready. Offspring. He ended up getting tall, short, tall. Let's total this up. So 3 out of 4 were tall. And then 1 out of 4, every 4 was small. Okay. And then he performed many similar experiments and came up with the following rules, which he called the laws of heredity. So Mendel's work led to the understanding that traits such as plant height are carried in pairs of information, pairs, in this case, genes, not by single sets of information. So, genetic traits are controlled by versions of genes that occur in plants. The two versions are called alleles. So you can have an allele, let's say, for a tall plant, or an allele for a short plant, or an allele, let's say, for brown eyes, or an allele for blue eyes. So what is the gene in this case that we've been talking about? What does it code for? It codes for height. And there are two versions. And later on, it can get more complicated, but we stick with the simple case of only two versions. That's the typical case, two versions of the gene. There's a tall and there's a short. And these two different versions are called, each of them are an allele. Because they code for the same basic trait, but the specific characteristic is different. They both of these alleles code for how tall a plant will be. The only difference is that one allele codes for a tall plant, and the other allele codes for a short plant. So we're going to use symbols just so we simplify this. A big T for a tall plant and small t for a short plant. This is typical of genetics. We use an uppercase to designate dominant. This is 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 domin That's the dominant allele. This here, I don't have enough room, this here is the recessive. Allele. Okay, so down here it says one allele always hides or masks the presence of the other, and this is called the principle of domination. And which one is the dominant allele? It's the allele that's always expressed and hides others. Okay, so if it's there, it's the one that's going to get expressed. It will mask the other alleles. The recessive, in this case, we're talking about the height of plants, a short plant, a short allele. The recessive allele is the allele or gene that, I'll put for gene, that's only expressed, not always expressed, only in specific cases, only expressed when the dominant gene is not present. The dominant allele or gene. So alleles are separated during, you already know about this from Science 9. I believe it's Science 9. Curriculums have changed. Oh, we already know that there's a separation that occurs during meiosis, and specifically during the first phase or stages of meiosis called meiosis 1. And this is called the law, when they're separated, it's called the law of segregation. They're separated. And they're mixed. That happens so that we can get a mixture of alleles when they come back together between the male and the female. And that way we get different physical characteristics. They're not all going to be tall. They're not all going to be short. Some of them will be tall, some will be short. Okay, so we're going to use a tool to determine how many of the different physical characteristics we're going to get and the different gene types. So this tool is called a Punnett square. We're going to use that square, if we go back here, to predict this, what I was talking about earlier. Okay, so this is the first generation. We're going to use the Punnett square to predict these numbers, the 3/4 and the quarter. How do we get that? Given the alleles. Okay. Okay. So we don't simply have to stick with tall and short. We can talk about other characteristics. So just to keep in mind, the letters are used as symbols to designate genes. So in other words, up here, big T designated the dominant allele, lowercase t designated the shorter recessive allele. Okay, so we can use letters to designate these traits. We're going to use, now we're going to talk about different color hairs. So you could be a redhead, red hair. R could stand for a recessive blonde trait. This doesn't necessarily have to be humans, it could be any species, but capital letters, go back to here, capital letters are used for dominant genes, lowercase are used for recessive genes, for traits that don't show up as often. They are recessive. Genes always exist in pairs. Okay, so you may have, for example, you may have the recessive gene for blue eyes, but it's not a dominant feature, so it didn't show up, and it may show up in your offspring if you marry someone with another recessive allele that codes for blue eyes. Okay, so let's go to this Punnett square. How does it work? You take this letter and you combine it with this letter, and you get that pair, which stands for what? Red. Two red alleles, so it has to be red. Okay, next, let's move over to the right. Combine this with this, you get capital R, lowercase r. Now, what's the overall color here? It's not a blend. It's not a blend between red and blonde. It's determined by whatever is the dominant allele. In this case, red hair. So this person turns out having, even though they have a combination of alleles, they end up having red hair. Okay, now next, let's go down to this third coordinate here. How do this combine the lowercase r with the uppercase r? What color does that determine? Red hair again. Next, let's do it again. We get this combined with that. What do you get? R to the recessive genes, which means you're going to get a blonde. You get blonde hair. Okay, so this explains the numbers that you could actually predict. 3 out of the 4 are red-haired, and 1 out of the 4 is a blonde. Okay, so let's just write down some more definitions. Lots of definitions in biology. So if you got a good memory, you do well in biology. Genotype. This set of genes possessed by an organism. Okay, so let's go back to this Punnett square. What are the genotypes? That's a capital R genotype. That's a capital R, lowercase r genotype. And this here is a lowercase r genotype. Those are genotypes. Okay? Now, genotypes and phenotypes are two different things. Phenotypes are what you actually end up looking like, what the physical characteristic is visually. Not what the genes are, but what the genes produce. Phenotype. You don't need that. Okay, so these are the physical characteristics an organism it has. And what does it depend on? Well, it depends on this. It depends on the genotype. The genotype determines the phenotype. Okay, the genotype determines the phenotype. And that's basically what we were saying here when we were drawing the Punnett square, or when we were going over it. Okay, so let's go down here. So a widow's peak is dominant. Let's look at some other features here. Now we're going to look at a widow's peak. Dominant, and it would be symbolized by W. Well, a no widow's peak recessive allele would be symbolized by a lowercase w. So let's say we have a father with no widow's peak. So let's say the father is WW. No widow's peak. There's no dominant allele there. It's just too recessive. The mother has a widow's peak. So she could be WW or W, capital W, or capital W, lowercase w. Let's make her WW. Sorry, it's confusing. Let's make the mother this. Capital W, lowercase w. Okay? Let's take a Punnett squirrel to determine both the genotypes and the phenotypes. Genotypes first. Phenotypes next, or second. Okay? So what do we do here? What do we put on the, or in this area here? Right, well, we write down, we can put the mother or the father, doesn't matter the order, but we'll write down the genotypes. So that we start off with, we have for the mother, W, uppercase and lowercase. And then what do we put here? Put the father, whatever's remaining. In this case, lowercase w, lowercase w. And then we just match them. So we match this, and we get W uppercase, W lowercase. And then again, similar, we match this, and then what do we get? We get lowercase w, lowercase w. And then we go over here, match this with this, and we get uppercase W, lowercase. And then we do it again, we match that with that, and we get lowercase, lowercase. So the question is always, what are the genotypes, and then what are the phenotypes? So the genotypes, and how many of each do you have? Okay, there's really only two genotypes. You got capital W, lowercase, and then lowercase, lowercase. How many of the genotypes are uppercase, lowercase? 2 out of 4, or 50%. What about lowercase, lowercase? 2 out of 4, same, 50%. 50% of the genotypes are lowercase, lowercase. Okay, next question. Next question is always, what's the phenotype? Well, what does this give us? Let's go back over here. What does that give us? There's one dominant allele, and therefore that means that 2 out of the 4 genotypes are widow's peaks. So, 1. Widows. How many? 50%. 2 out of half. What about no widows? No widow peaks. 50%. Because lowercase, lowercase means no widow's peak. Okay, let's go back to definition. Lots of definitions. You need to know the definitions. They're homozygous. That is a definition for 2 copies of the same gene. Example, uppercase, uppercase, or lowercase, lowercase. 2 copies of the same allele or genes. Okay, so an example is WW, uppercase, uppercase, or lowercase, lowercase. Those would be homozygous pairs. Okay, what about heterozygous pairs? Two different genes or alleles. So example of that is uppercase, lowercase. Those are heterozygous pairs. Okay, so classify whether the genes are homozygous or heterozygous. So the first one is homozygous. And we would say, since they're both dominant, we would call this homozygous dominant. Next one, they are heterozygous. And you don't have to say that whether they're dominant or recessive. Doesn't make any sense in this case. In the next case, it does. These are obviously the same. They're both the same. We use the word homo for meaning the same, homozygous. And they are recessive. They are not dominant alleles. So we label this homozygous recessive. Okay. Okay, just read the next portion there. So we have monohybrid crossing involves only one gene pair for each trait. Okay, so this is the easiest thing. I mean, it gets more complicated later on, but we're going to start off with monohybrid crossing. Okay, so let's cross a homozygous dominant. For example, BB, or B stands for brown eyes. Gene is, or allele, gene or allele is gene is eye color. So B stands for brown eyes. And lowercase b stands for blue eyes. So uppercase is brown. Okay. So homozygous dominant to brown, and the recessive is blue. We know that from experience as less blue-eyed people. So we label lowercase b. Okay. So what is the genotype and phenotype make up the Punnett square? That's what we're trying to answer with the Punnett square. That could be one of your questions you make up, right, in this information. So what we write here, I could write B, uppercase, uppercase if you want, or lowercase, lowercase. Let's do lowercase, lowercase. You just copy this here, or this. It doesn't matter which one you put on the side or on top. I'll put them in the correct order. So B, B on top, and match them. You get B, B here. You get B, B here. They're all B, B. One is capitalized in the first two. And the third one was capitalized. The dominant cap first. And then the last one is capitalized. So there's a dominant gene in each of these pairs. This is called the F1 generation. I think we talked about that. First generation, F1 generation. Okay, now the question in all of these cases where we're dealing with crossing is, what are the genotypes and phenotypes? So the genotype first, because that determines the phenotype. Well, they're all capital B, lowercase b, so it's 100%. B, B. What's the phenotype? Well, the genotype determines the phenotype. Phenotype is what you see outwardly. What's the outward appearance? So 4 out of the 4 are brown eyes, 100% brown eyes. Okay. Okay, F1 means first generation. Now, let's see what happens when we cross the following. Two F1 generations. And how do we write that out? Uppercase, lowercase, times uppercase, lowercase. The question, what are the genotypes? What are the phenotypes? Again, come up with a Punnett square. We put capital B here, lowercase, capital B here, lowercase. And then just cross them. So you get B, B, uppercase, 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 lowercase here, uppercase, lowercase, and then lowercase, lowercase here. So what are the genotypes? And then what are the phenotypes? Okay, B, B, make a list. You have B, B, capital B, capital B, capital B, lowercase b, and only 3 types, lowercase b, lowercase b. How many of each? Well, 1 out of every 4 is capital. Capital B, capital B. 2 out of 4 is uppercase and then lowercase b. And then 1 out of the 4 is lowercase, lowercase. So what does 1/4 translate into? 25%. It's a quarter. You just define the percentage, multiply the fraction, a quarter by 100, 25%. 2 out of 4 is a half, times 100% is 50%. And again, here, this is 25%. Okay? So what do we call this generation? This is no longer F1. It's a combination of F1s. So this is called the second generation. This is F2, or you could call it second generation. That's what it means. Okay, what are the phenotypes? You always have to find the genotypes first. Okay, well, which one's brown eyes? This and this. Even though there's a recessive, this dominates. So that means 50 plus 25, 75%. Brown eyes. 3 out of 4, 75%. What about blue? Blue-eyed people, in this case, that turned up only 25%, 1 out of every 4. Okay, memorize these. I would go over these. All of them, yeah. And that's it, guys. Well, I've got more to give you. I've got one more, I think one more lesson. So I'll do that in the next few days.